Hey guys, Miss Cote here, and this is the next video in the Cell Communication and Cell Cycle unit. In this episode, we're going to start looking at how the cell cycle is controlled and what happens when the cell loses control of the cell cycle. So let's get started. In the last video, we looked at the phases of the cell cycle. We saw that when a cell is not dividing, it's in interphase, which is about 90% of the time. When a cell does divide, it goes through a series of stages called prophase, prometaphase, metaphase, anaphase, and telophase to divide the nucleus and the DNA. Then the final step would be cytokinesis, where the cell divides the cytoplasm and physically splits into two new daughter cells. Each daughter cell is genetically identical to each other and to the original parent cell. Now comes the new material for you guys, how all this is regulated. Remember, regulation of different processes is very important in understanding biology. We have to know what something is going on in a process in order to understand what happens when something goes wrong. How often a cell divides depends on what type of cell it is. Prokaryotic cells divide a lot quicker than eukaryotic cells, since they're so simple. And even among our own body cells, some cells multiply more than others. Some of our cells, like mature nerve or muscle cells, never divide. The ability to divide or not comes down to regulation at the molecular level. And when our body loses control of cell division, that's when cancer comes into play. Cancer cells are cells that grow out of control. They no longer follow the control of the cell cycle. As scientists search for an explanation of how the cell cycle is controlled, one of the leading thoughts was that maybe there are chemicals in the cytoplasm of the cell that act as signals. One of the experiments that supported this was to take two cells at different stages of the cell cycle and fuse them together. Fusing a G1 and S cell together prompted the G1 cell to enter into the S phase and start replicating its DNA. Since G1 and S are adjacent stages, maybe the G1 cell was about to enter into S anyway. To further prove their point, they fused a cell in G1 and a cell in the M phase. And immediately the cell in G1 started mitosis, skipping the S and G2 phases completely. This suggested there were molecules in the cytoplasm that control when a cell moved from one phase to another during the cell cycle. That experiment, along with others, demonstrates that there is a cell cycle control system that coordinates the timing of events in the cell. There are specific checkpoints during the cell cycle where if the cell is not ready to move on to the next stage, the checkpoint will not allow it to proceed. If a cell that was damaged in some sort of way, let's say the DNA was not replicated properly, if that cell was allowed to divide with damage, then its daughter cells would be damaged as well, and so on and so forth. So here you can see that there are three major checkpoints along the cell cycle, represented by the red gates. One toward the end of G1, one at the end of G2, and another during the M phase. These checkpoints are controlled by both internal and external signals. But before we go into the checkpoints, let's just look at those cytoplasmic molecules that very simply control a cell's progression through the cycle. Then we'll go more into what happens at each checkpoint. The molecules in the cytoplasm that control the cell cycle are two proteins, cyclins and cyclin-dependent kinases, or CDKs. If you haven't already figured it out by the ASE at the end of the name, the kinases are enzymes. So here's the deal. The CDKs are always present in the cell. It's the level of the cyclins that change over the course of the cell cycle. And when cyclins are present, they form a complex with the CDKs. And these complexes are called maturation promoting factor, or M phase promoting factor, or MPF for short. So basically, the more MPF that is in a cell's cytoplasm, the further it is in the cell cycle. Let's put it into context by looking at a diagram. This graph, and hint, hint, you will see this graph again, shows the changes in the levels of MPF, the orange line, over the course of the cell cycle. Notice that the increase in MPF corresponds to the increase in cyclins, the purple line. While the cyclins start gradually building up during the S phase, toward the end of G2, we see a sharp increase in MPF, which pushes the cell into the M phase. Once in the M phase, there's a sharp decrease in cyclins as they start to be degraded, and a decrease in MPF follows. What's not shown in the graph is the concentration of the CDKs. If we were to draw a line representing the CDKs, it would be a straight line, since the concentration of the enzyme stays constant. It is the cyclin concentration that fluctuates. Here's another way to look at it. When the new cell first starts out, the cyclins are finishing up being broken down, but the CDK remains. Remember, it's constant. The cyclins don't begin to be made again until late in the S phase, after the DNA has been copied, and they start accumulating during the G2 phase. Now they pair up with the CDKs to form the complex called MPF. When there is enough MPF in the cytoplasm, then the cell passes through the G2 checkpoint and the cell will start mitosis. Once anaphase happens, passing through the M checkpoint that is not shown, then the cyclins will start being degraded and the cycle will start all over again. So that takes care of the cytoplasmic molecules that act as signals for the progression of the cell cycle. Now let's look more closely at what happens at the different checkpoints. Let's go back to this slide from before. Remember that the three important checkpoints fall during the G1, G2, and M phases. Internally at each checkpoint, the cell basically gets examined to make sure everything is working correctly and is ready to proceed to the next step. There can also be external signals to determine whether the environment is right for the cell to divide. So what needs to happen at each checkpoint so that the cell can continue through the cycle? For most cells, the G1 checkpoint is the most important because this is where the cell will get the first go-ahead to ultimately divide. Remember earlier, I said that different cell types will divide at different rates. Some go through the cell cycle quicker than other types of cells, and some never divide at all. Well, here is where that comes into play, at G1. Now, I don't know why this part is all the way over to the right, but if the cell gets the go-ahead signal, then it will continue on to S, G2, and then M. But if the cell is not ready to divide, it can exit the cell cycle and switch into a non-dividing state called G0. Now, just because it goes into G0 doesn't mean it will never divide. When the time is right, a cell can come back into G1 and eventually divide. But some of our cells, like mature nerve and muscle cells, never divide. The cell moves into G0 and stays there until the cell eventually dies. The next checkpoint would be at the end of G2. We said earlier that the cell will not pass through G2 unless there's enough MPF in the cytoplasm. And this correlates with having enough energy for mitosis. And not that they show it here or talk about it in the book, but most likely one of the other important factors to pass through the G2 checkpoint would be if the DNA has copied itself correctly and there are no errors or mutations. I think that's pretty important thing to consider. I wouldn't want any mistakes in the DNA being passed on to future cells. During the M phase, while the cell is in the process of dividing, scientists have worked out that the checkpoint is there to make sure all of the chromosomes are attached to spindle fibers through the kinetochores. Only at that point will the cell begin anaphase. It wouldn't be good if the cell divided and ended up with either missing or extra chromosomes. So not only do you need to understand the correct order of the cell cycle and what happens at each phase, you should also understand that this whole process is under regulatory control. The cell just doesn't do whatever it wants to when it wants to. There are internal signals that keep it under control. As far as external signals are concerned, through various experiments, such as this one here, scientists have found that there are certain molecules called growth factors that stimulate cells to divide. Our example here is using a growth factor called platelet-derived growth factor, or PDGF. Cells can be grown in culture as long as they have the nutrients they need to live, just like we need nutrients to survive. However, you can see that with the addition of PDGF, the cells not only live, but also multiply, as opposed to the cells without the PDGF. There are many different types of growth factors found in our cells. This is just one example. Another external signal is the availability of space to grow and a foundation to grow on. As shown in this diagram, in order to divide, most cells need a foundation or base to grow on, to anchor themselves to. This phenomenon is called anchorage dependence. In cell culture, this surface would be the bottom of the flask. In your body, cells grow on extracellular matrix. And these cells will continue to grow and divide until they form a single layer and there's no more room. This is called density-dependent inhibition. When a cell touches another cell, this is a signal that it's getting crowded and it will stop dividing. In the case of a wound, if some of those cells are removed, the cells will divide to fill in the space and then stop when they touch. Between anchorage dependence and density-dependent inhibition, this keeps cell growth in check. Unfortunately, sometimes a normal cell may be altered or transformed in some way to become a cancer cell. Cancer cells do not respond to normal cell cycle controls. Everything you just learned about how the checkpoints and controls of the cell cycle is thrown out the window. Basically, cancer cells just do what they want, and more often than not, they like to grow out of control. For example, here they do not recognize anchorage dependence or density-dependent inhibition. They're growing on top of each other, forming a clump of cells. This is how tumors are formed. Please know that we do have an immune system that is trained to recognize transformed cancerous cells as not being normal, and there are mechanisms in place to destroy them. But nothing is perfect, and sometimes these cells go undetected and allowed to grow uncontrollably to form a tumor. There are two types of tumors, benign and malignant. You may have heard these terms before. While tumors are clumps of cancerous cells, benign tumors are confined to the original site. Usually, surgery is done to remove the clump of cells, and the person may have to undergo a round of radiation to kill off any cells that may still remain. The bad types of tumors are the malignant ones, because cells can break off of the original tumor and metastasize or travel to other parts of the body. This is when the cancer spreads. Once they settle in somewhere, they can grow into another tumor. Chemotherapy is usually used to treat these types of tumors. These are drugs designed to interfere with specific steps in the cell cycle and kill actively dividing cells. Unfortunately, you also have cells in your body that are continually dividing, which is why some of the side effects are hair loss because of your follicle cells, nausea because of your intestinal cells, and you're more than likely to get an infection because of your immune cells. Due to all of the cancer research that's been going on for years, scientists are getting better at treating different types of cancer. Unfortunately, there's no one cure for cancer. Each type of cancer seems to be different, and we're finding that while one type of treatment works well for some, other cancers require a different approach. And that brings us to the end of this chapter. If you look on Google Classroom, I've posted a link to the cell cycle control game, which is a fun little way to review not only how the cell cycle works, but also the checkpoints that are involved in making sure the cell is ready to move on to the next stage. And until I see you next, be a good human.